should begin. So we leave a little time for the psychiatry. I believe that's to follow. By an amazing coincidence, I just received a chapter on glycogen storage diseases for the 7th edition of the Metabolic Basis of Inherited Diseases, of which I'm an editor. It was the last edition, the 6th edition, was published in 1987, and the new one, or maybe it's 88, and the new one will be out next year. And a new writer wrote the chapter on glycogen storage, and there are a number of new things that I'll mention to you, which are not yet in the textbooks. They might not even be in the next edition of the textbooks, but they increase our understanding of the topic that we're discussing today. I'll try to finish in time for the debates. When do the debates begin? 8 o'clock this evening. Okay. Good. So on completion of this conference, the lecture objectives were to explain the nature of the biochemical defect in type 1 glycogen storage disease and the specific manner in which this causes problems in fat and carbohydrate metabolism. That's kind of interesting, and you'll reflect on this clinical correlation after you study fat metabolism, which comes next in the sequence, because this disturbance in carbohydrate metabolism shows you an interesting interplay between fat, carbohydrate, and amino acid metabolism. Second objective is to recognize the effects of epinephrine and glucagon on carbohydrate metabolism and how these challenge doses are used in diagnosis of these specific disorders. Third is to recommend a satisfactory treatment. Fourth is to recognize the cause and methods of managing hyperuricemia in von Gerke's disease. And later we'll have a clinical correlation on gout, which is the clinical consequences of hyperuricemia. And again, this will be relevant to those. The metabolic discussion here today is relevant to that topic as well. So first I'll begin with the case that we have before us, the case that was presented. Then I'll show you some of my own, in my own experience. Well, actually, before we begin the cases, I think I'll just review briefly the glycogen pathway here, which Dr. Longmore discussed with you. Pathway of glycogen breakdown. Now, it turns out glycogen storage diseases can result from disorders of glycogen breakdown and also from some disorders of glycogen synthesis. And there are Roman numeral numbers which go beyond the number which I can remember. And the numbers are like the clotting factors. They don't have any significance except that they were numbered in the order with which the disease was discovered. So the first one that was discovered is called glycogen storage disease, Roman numeral 1. And the second one, which has nothing to do with that, is Roman numeral 2, just because it's the second one that was discovered. Let's see. I think I'll have that first slide. Great. Okay, so this is just a picture of the glycogen molecule, which should be familiar to you. Let's see if I can focus that just a little bit. There we go. Okay, the glycogen molecule, which is a highly branched molecule that serves as our primary store of glucose for energy in liver and muscle. And one thing that we know that's new about it, I think Dr. Longmore said that there's some evidence now that it's synthesized originally on a protein, and now we know exactly that it's synthesized on a protein, and that the synthesis of this glycogen molecule takes place on a 37,000 molecular weight protein called glycogen, which has a tyrosine, a specific tyrosine that is glucosylated. And 2 glucoses are added to that tyrosine, and then this glycogen molecule actually is autocatalytic and is able to actually add 6 more, and that then becomes the primer for glycogen synthesis in liver and muscle. And what happens is that glycogen molecule with its 8 sugars combines with one molecule of glycogen synthase, and the glycogen synthase then adds all of the subsequent the subsequent 1,4 linkages that are added to this molecule. And as Dr. Longmore explained to you, there is a second enzyme that participates called the branching enzyme, which can take a minimum, I think, of 6 glucose residues and transfer them from a 1,4 linkage to a 1,6 linkage, so that the 1,4 is still free to grow, and we wind up with this enormous multi-million molecular weight compound, which contains primarily long polymers of 1,4 linked glucose residues with branches having 1,6 linkages. When the process is reversed in the liver and muscle, as he explained, it's done by a different set of enzymes. The phosphorylase moving off the 1,4 residues one at a time, producing glucose 1-phosphate, and then requiring a different enzyme. When it nibbles down to a chain length of 4, then it requires a different enzyme, the debranching enzyme, to remove these 3 glucoses and this 1,6. Now we know that in fact, that those 2 activities are part of one enzyme. One 170K protein has those two different catalytic sites that catalyze those 2 activities. And we also know that one of those 2 catalytic sites now can go the last mile and take it right down to the tyrosine residue from the glycogen protein molecule. So this whole process, the ones that you'll read about in the textbooks are the debranching enzyme, and the debranching enzyme has 2 activities, one of which is the glucosidase activity, and the other, the transferase activity, and the 1,6-glucosidase activity. Now this allows the release of some glucose molecules from the degradation of glucose, even if we can't dephosphorylate glucose 6-phosphate. So what's the primary product of phosphorylase reaction that removes all of these glucoses? Okay, so the primary product is glucose 1-phosphate, and for that to become glucose, it has to be converted to glucose 6-phosphate, and the glucose 6-phosphate has to be cleaved. What's the product that's released when these 3 are released? Glucose 1-phosphate, right? And the product when this is released is glucose, right? Okay, good. Okay, so this is just a picture of that reaction. Phosphorylase releasing the glucose 1-phosphates, and the glucan transferase and the glucose amylo 1,6-glucosidase, our debrancher enzyme, being the same, being a different catalytic site, but the same enzyme activity. So this is the pathway. This is the storage molecule, the gigantic molecule that began its life being synthesized on a protein and 8 oligosaccharide primer and assembled into this gigantic molecule. We're not really going to talk about the biosynthetic pathway significantly, but that molecule serves as a glucose store in liver and muscle, and the release of that glucose requires the phosphorylase reaction, the amylo 1,6-glucosidase to a less extent, and production of glucose 1-phosphate, which then has to be converted to glucose 6-phosphate, and this in turn has to be converted to glucose. Now, one important thing about this reaction, where does all this take place in the cell when we think about compartmentalization? It takes place in the cytoplasm of the cell, right? Where does this reaction take place? Probably wasn't covered in your lecture, but that reaction takes place. The glucose 6-phosphatase the catalytic subunit of the active site of glucose 6-phosphatase faces the interior of the endoplasmic reticulum. Okay? So in order for this glucose 6-phosphate to be hydrolyzed, it has to be transported into the lumen of the endoplasmic reticulum, and then that phosphate is cleaved, and that phosphate, the cleavage of that phosphate then would release glucose. Now, glucose then has to get out of the endoplasmic reticulum in order to get into your bloodstream and be available. And this is a picture of what happens. Glucose 6-phosphate is transported into the thing that Dr. Longmore, I'm sure, stressed is that phosphorylated sugars don't easily move across membranes, and they have to be transported. So glucose phosphate has to be transported into the lumen of the endoplasmic reticulum. And this slide illustrates a couple of other things, that there are products of this reaction that are formed that also have to get out. And the reason that they have to get out is because if all the glucose were hydrolyzed and the products that were the phosphates that were released were not able to escape, this reaction would soon become product inhibited. And so in order for that to get out, these are also charged molecules which have to go across membranes. These also require a transport mechanism to get across that protein, to get across that membrane, rather. And in fact, the glucose itself, although it isn't drawn here, itself requires a glucose transporter. It's called GLUT7, a recently identified glucose transporter, which allows the product of that reaction to get out. Now, what are the implications of that? How did we discover that? Well, we discovered that by finding out that patients who looked like the little patient that was presented to you today don't all have the same disease. What was called glycogen storage disease type 1, which is what's presented to you today, actually turns out to be a mixture of diseases. Most of the patients are in fact missing or defective in the glucose 6-phosphatase. But in fact, there are 6 proteins that are involved in this conversion. And now we're finding a rare patient who tells us, we learn about each of these proteins by finding a rare patient who's missing one of those. So there are in fact the main patients with this disorder which are missing or have defects in this enzyme, which is the glucose 6-phosphatase enzyme, which has now been cloned, and we know, it's now been purified, and we know what the catalytic unit is, and the catalytic unit is a transmembrane protein that faces the interior of the endoplasmic reticulum. There's a second protein whose function we don't fully understand, called the stabilizing protein, which associates with that in the membrane, and some patients have this disease who are missing the so-called stabilizing protein. And then we have found that some patients have this disease who are missing the transporter. And that now is called glycogen storage disease type 1b. And it looks exactly like the patient who was presented today, with one additional feature. And that is that they have very low white blood cell counts, telling us that this transport protein has some important function in white blood cell maturation. We don't understand it yet, but they look exactly like the patient that was presented today, but they have very low white blood cell counts and they're susceptible to infection. And it turns out now that there's a glycogen storage disease type 1d, which is missing this transport function. And there hasn't yet been discovered a disease, but there is a protein which is required for transporting glucose out of the ER, and it's likely that we'll find somewhere along the line a glycogen storage disease type 1, A, B, C, D, E, which is probably missing that glucose transporter. So there are 6 proteins that are involved in a simple, in this reaction, and we've learned about each of them, except this glucose transporter, through a storage disease that told us what they, what, yes?
Patients need to be homozygous for the defect?
Yes, that's a good question. Almost all of the glycogen storage diseases are autosomal recessive, requiring that you inherit the defect from both parents to be completely absent. The exceptions are the liver phosphorylase enzyme. I think not liver, one of the protein phosphatases. There's an X-linked glucose 6. I'm sorry. Glycogen storage disease type 6 is an X-linked disease, and so males only have one copy of that gene, and they show X-linked inheritance. So in all the others, the inheritance is autosomal recessive. Okay, so let me see. I think are there any questions about this? So this patient that we presented today has glycogen storage disease type 1, period. Okay? But in fact, when you're on the wards, it turns out that that could represent one of several different missing proteins. If you had a very low white blood count, you would suspect type 1b, which is the missing protein which transports glucose 6-phosphate into the endoplasmic reticulum. There's another reason. The way this was discovered was not that way. It was actually discovered in another way. Let's see. It turned out that there were patients who looked exactly like the patient that was presented today, who had glycogen storage, had the clinical phenotype for glycogen storage disease type 1, who had liver biopsies. And when the liver biopsies were measured fresh, they had no activity, right? When they came back and measured them somewhat later to verify that enzyme defect, they found glucose 6-phosphatase activity. So what happened? Right? They stored them in the freezer, they came back and they looked at them and they found glucose 6-phosphatase activity. Well, that's the answer, that they stored them in the freezer. Because in the freeze-thaw, they broke open the endoplasmic reticulum and exposed the glucose 6-phosphatase, which was normally present inside, and overcame the defect of the transporter, which is present in the membrane, right? So the important thing is that you have to measure this enzyme either in freeze-thaw preparations or in the presence of detergent in order to detect the glucose 6-phosphatase in type 1b. If you measure it in fresh tissue without detergent, and that's in fact what one does, if one is looking for the enzyme activity in a liver biopsy specimen, one measures it in the presence and the absence of detergent. So if I had type 1a and I added glucose 6-phosphate and I looked for the production of release of phosphate, I had type 1a and I looked with and without detergent, what would I find? Wouldn't work in either test tube, right? But if I had type 1b, I'd get no activity in the absence of detergent, and I'd see activity in the presence of detergent, right? And that's how type 1b was discovered. And then there was the correlation made that all the patients that have type 1b are the ones who have this very low white count and the susceptibility to infection. Okay? So that's what we call delineating genetic heterogeneity. Here we have look-alike diseases that are actually due to mutations in different genes and therefore defects in different proteins, and we sort them out by this kind of approach. Okay, so the patient today is a Caucasian male, 8-pound, 4-ounce product of a normal full-term pregnancy, first child of a 21-year-old father and 18-year-old mother who were first cousins. Why is that relevant? Relevant to the question he asked, do they need to be homozygous for this condition? And first cousins are more often homozygous for a rare trait than population at large, right? Because they share 1 out of 8 genes at any one locus with each other.
So heterozygous
Heterozygotes show no symptoms for any of these disorders. That's correct. So at birth, the infant was noted to have a slightly enlarged abdomen. At age 1 month, he had generalized convulsions, but recovered. At age 4 months, he had a second series, was admitted to the hospital for evaluation. And then that time, he was noted to be below the 3rd percentile for length and 10th percentile for weight. Now, what do percentiles mean? Every time you get a baby in the hospital, after you measure them and weigh them and so on, you go and you plot them on growth charts, and the growth charts show you what the 3rd to the 50th to the 97th percentile of normal growth, usually the first 24 months, and then there's a separate chart for beyond that. And so this baby plotted out below the 3rd percentile. That means 97% of children the same age or bigger. And the 10th percentile for weight. Had a greatly distended abdomen, which they show you a picture of in that, and I think I have a picture of a real patient with the same problem. Okay, this is a real patient, and he didn't come on admission with these marks on his tummy. They were made by the physicians who were trying to demonstrate for this photograph the size of his enlarged liver. And that liver completely, you see this is the right lobe and the left lobe of the liver, which completely fills the abdomen of that child who has glycogen storage disease type 1A, like the patient presented today. So also noted on the patient presented today were several xanthomas, which aren't apparent, I don't see in this picture. Xanthomas are cutaneous fat deposits and a slight odor of acetone in the breath. And the reason there's acetone in the breath is because there's there's an odor of acetone is because there is acetone in the breath. We breathe off acetone when it's present in the serum. Other lab values were listed, and those are in your handout there. There are several striking abnormalities in the lab values. One of them is the fasting blood sugar. Fasting blood sugar is 14. If you or I had a fasting blood sugar of 14, we wouldn't do well on our exams. In fact, we'd probably be convulsing, because that's profound hypoglycemia. And this patient, however, was not at that time convulsing. And as is explained later, it's because these patients develop a resistance to hypoglycemia, which we'll talk about later. The other thing is the blood lactate level was extremely high. Where does lactate come from? From glycolysis, right? And so we've got fully charged, turned on glycolysis. Blood pyruvate is also elevated. Less obvious to explain is elevated serum cholesterol of 600, which for a child this age is an enormous elevation. It would be a big-time elevation for an adult. Phospholipids and triglycerides are very high, and serum uric acid is 9, which is normally 2 to 5 for a child this age. And the total ketones are considerably elevated, as are the free fatty acids. Okay, now they did a glucagon and galactose tolerance test. You talked about galactose tolerance tests last week, probably in galactosemia, right? Okay, and this one, which is given in your handout, we have the patient on the left, and the normal infusion of galactose, just to show you that it got in, right? The galactose elevation is prompt, right? And is this the glucose rise? No, that's not the glucose rise. The glucose doesn't budge, but instead what happens is the lactate is dramatically elevated. Okay, so that's an abnormal galactose tolerance test. Would that be the kind of galactose? How did that resemble the galactose tolerance test in the child with galactosemia? That was abnormal also, and I don't remember the I don't know that lactate was measured in that test. But anyway, here's the normal control, and what you see here is the galactose rises, then the galactose is assimilated. The rise in glucose represents the normal conversion of galactose to glucose through UDP gal and UDP glu, which does not occur in the galactosemic patient that you saw last week, right? So the galactosemic patient that you saw would not have this rise in glucose, right? And the lactic acid doesn't rise, doesn't budge at all, because most of this galactose is getting converted to glucose, which is not being metabolized through the glycolytic cycle. So the galactose, yes?
Last week, Dr. Schumacher mentioned that the swelling of the liver is due to tissue damage, but he didn't know what the damage in the,
I see.
in the liver was from the galactosemic.
Yeah.
So what is it?
Okay. The question is, Dr. Shoemaker said last week that kids have liver trouble in liver enlargement with galactosemia, and these kids have liver enlargement, but they're totally different. In the case of galactosemia, it's thought that the toxic product is gal-1p, which is present in too large a concentration, and what these children actually get is a toxic necrosis of the liver, and they develop cirrhosis. They get very serious cirrhotic changes. These children accumulate large amount, and in fact, I think, isn't there a biopsy here in this one? They say they biopsied the liver, and what they found was excess glycogen of normal structure. Okay, and that's of normal structure, excess glycogen of normal structure, and that's diagnostically important. So the other thing these kids have is a large amount of what else? They tell you in here that the liver is full of glycogen and fat. They have a fat storage disease as well as a glycogen storage disease. And that's because they're over-synthesizing fat from the glycolytic products, right? Okay, so they don't have cirrhosis of the liver at this age. We will discuss one little patient, if we get to it, who does in the glycogen storage. Yes?
Are these patients acidotic with all this lactate?
They are, in fact, certainly the patient was said to be acidotic after treatment, and I think, in fact, they are mildly acidotic, right? This patient later was treated and was acidotic during the treatment, and they mentioned that they gave bicarbonate in order to control the acidosis. Other questions before we go on? Okay, so the galactose tolerance test was abnormal. We know why is it abnormal? Because galactose, in order to be, does the galactose get converted to gal-1p? Excuse me, all the galactosemic pathway that you studied last week is normal. Right? What's abnormal here is that the product, the glucose 6-phosphate, which it has to go through, can't be degraded to glucose. And as a consequence, all the galactose that's administered gets metabolized through the glycolytic pathway, and that explains the elevated lactic acid. Okay, the glucagon test is abnormal. Now, if I gave you glucagon or you gave me glucagon, just to be fair, what would be the expected response? Pardon? Expected increase in blood sugar. And why do you get an increase in blood sugar? Because you mobilize glucose from glycogen, right? And why can't these patients, how is this patient's glucagon test abnormal? It's flat instead of showing hyperglycemic response to glucagon. And the reason it's flat is because it's for the same reason. Now, what would be the lactate response to glucagon if you gave this patient glucagon? You'd expect you'd expect if you mobilize glycogen, but you can't metabolize it, that in fact it would go through the glycolytic pathway and you'd get lactate production. Okay. Now, so why is the child hypoglycemic? We have a lot of abnormal things to explain here. We have an extremely low blood sugar. Why is the child low in blood sugar? Well, what are the two sources of blood sugar, of raising your blood sugar? If you had your if I gave you two chances to raise your blood sugar and you could use either ones, which would they be? Be either gluconeogenesis or mobilizing it from glycogen, right? Do they both require glucose 6-phosphatase? Right? They do, in fact. In other words, to mobilize glucose through gluconeogenesis, to produce glucose through gluconeogenesis, you have to go through glucose 6-phosphate. To produce it from glycogen, you have to go through glucose 6-phosphate. And so those are really the only two substantial ways of raising your blood sugar. And consequently, this child, after a feeding, its blood sugar rises normally from alimentation, right? But subsequently, after 3 to 4 hours, blood sugar, when that alimentary load is cleared, then the ability to maintain the blood sugar requires our ability to either make it from gluconeogenesis or recover it from mobilize it from glycogen. And this child is unable to do either. So fasting blood sugar means that the child hasn't eaten in a number of hours, and the child, therefore, is profoundly hypoglycemic because it can't produce glucose from either, significant amounts of glucose from either of those sources. Okay, so the diagnosis and treatment, the clinical impression was glycogen storage disease type 1, which was named after von Gierke, and the absence of glucose 6-phosphatase activity in the liver specimen confirmed the diagnosis. And the glycogen was present in elevated amounts but normal in structure. So if you got the answer from the laboratory that the glucose 6-phosphatase measurement was normal, what would you ask? You'd say, was that done in the presence of detergent as well as in the absence, right? And if it wasn't, then it ought to be reassayed in the presence of detergent to make sure you could distinguish glycogen storage type 1 and 1A from 1B. Okay, so now we know what the patient has, and so we begin our treatment protocol, which is explained there. And the goals of treatment, which are outlined in dealing with this patient, are to maintain blood sugar in a near normal range. That's one goal. And to provide medium chain triglycerides as another source of nutrition, which they used here. Frequent feedings of glucose, though, is very important because it restores the insulin, the normal insulin response. This protocol says that one of the reasons these kids are dwarfed or very small is because they break down excess protein and because they're hypoglycemic. But another important reason that they're small is because they're hypoinsulinemic, because they have profound hypoglycemia. And insulin is a growth factor. And so we need insulin to grow, and by raising the blood sugar toward normal levels, we normalize the insulin levels and get the growth factor response from the insulin in that way. Okay, so why do they go at all the trouble of rigging up this Wheatstone bridge? I haven't seen that since my sophomore physics course when I was in college. Why do they go to all the trouble with this nasogastric feeding during the night? Maintain blood sugar. The reason that maintaining blood sugar is important, why nighttime, you could do it by waking the child and frequent cornstarch feedings as well. The reason that you have to maintain blood sugar during the night and avoid prolonged fast is that once you begin to treat these kids, they become normally sensitive to hypoglycemia. I mentioned to you that when they present to you, they're really quite resistant to hypoglycemia. And you know why they're resistant to hypoglycemia? Because their brain has become adapted to using other energy sources, the fatty acids and the ketone bodies. But once they de-adapt by normalizing their blood glucose If you then expose them to the same hypoglycemic shock, then they can suffer severe brain damage. And so that's the reason that one treats these patients with nasogastric feedings during the night. Okay, now, how good is the treatment? Well, this is a 11-year-old boy, I believe, with glycogen storage disease that's been following that treatment since childhood, and this is a kidney X-ray on a 23-year-old woman who's been treated since childhood with frequent feeding. And the purpose of this X-ray was to show her very large kidneys. These patients have large livers, but they also have large kidneys because their kidneys also store significant amounts of glycogen. But the treatment of this disorder has been extremely gratifying because frequent feedings, just cornstarch feedings, with or without the nasogastric feedings, has been dramatic in prolonging these children's life and normalizing their growth. And most of them now actually can achieve adolescence or adulthood without serious problem. A new long-range problem that's developing in these children is the development of liver nodules or liver tumors that develop because they live long enough to develop them. But the severe metabolic abnormalities are largely corrected by the approach that was used in this patient. And the treatment response, which I won't go through in this patient, in which they saw the glycogen stores come down, the hepatomegaly get better, the xanthomas kind of wilted away over several months, and the blood lipids cleared, showed the success of this kind of treatment. Okay. So, are there any questions about this particular patient or this particular? Yes.
They have to have the frequent feedings their entire life.
They get less dependent, but they always require frequent feeding. Actually, it's not so bad. Actually, cornstarch, every 4 or 5 hours is sufficient to maintain blood sugar in these patients. They become a little less dependent on frequent feedings as they move into adolescence, but they always have to avoid significant fasting. Yes? The hepatomegaly goes down somewhat. And it can almost completely disappear. Yeah, if they're ideally treated, it goes down quite substantially. I mean, usually you can feel a slightly enlarged liver, but the kind of picture that you saw in this infant is much improved. The 12-year-old boy I showed you, though, still had significant hepatomegaly and was, in fact, successfully treated. Yes?
Did they get to the point where they can, like, sleep through the night without having to get up and bed?
Yes. With cornstarch feedings, they can go about 6 hours, usually. I mean, a 6-hour is through the night, probably, huh? And so they can go 6 hours with cornstarch feeding. Okay. There were a couple of questions. Why do they develop problems? Why is the kidney enlarged in these patients? Because it's the other site of significant glycogen storage. What's the difference in the storage? What's the difference in the purpose of storage in muscle and liver? Why do we store in muscle, and why do we store in liver? In the muscle, we use it mainly as a substrate for local use, right? That's the main reason it's stored in muscle. And in the liver, we use it mainly to maintain, we use it significantly to maintain, to control our blood sugar level. So if you had glycogen storage disease in your muscle, would you be sick like the patient that we saw today? In fact, you would not, because you don't use that muscle glycogen to maintain your blood sugar predominantly. And the main features of muscle glycogen storage diseases, and there are several, are symptomatic muscle problems: cramps, fatigability, muscle wasting, weakness. Yes?
They're also fatal?
The muscle, well, let me show you a couple.
Just because
well-
Dr. Longmore had mentioned that all glycogen storage diseases are mentioned yesterday.
That's not quite right. There are some of them that are really mild, particularly type 5, which is called McArdle syndrome, which is muscle phosphorylase deficiency. It's not usually discovered until late adolescence. Now, in a way, he's kind of right, because if you have significant heart involvement, then these people frequently will develop premature death. He's probably right that the lifespan is slightly shortened, but people with muscle phosphorylase deficiency live quite at least into late adulthood. Okay, let me just run through. I brought some pictures of some of the others which illustrate some points. This is I'm just going to show you the other Roman numerals, and to make a couple points about glycogen metabolism. This is a let's see, I have those ages. 7 and 18. All right. This is a boy at age 7, mildly enlarged liver, and and the same boy at age 18, mildly enlarged liver, with glycogen storage disease, Roman numeral 3. Now, type 3 is a debrancher deficiency. Okay? Now think about it for a second and ask yourself, what would a patient who was unable to debranch his glucose, what kind of problem should he have? They don't get significant hypoglycemia usually. And why not? That's because they can whittle down their glycogen molecules quite a long way. And what would they accumulate in their liver? What would you, if you did a liver biopsy on this little guy, what would you find? You'd find glycogen present in excess, but they wouldn't tell you that its structure is normal. They would tell you that it has the structure of what's called limit dextrin, right? It can go down to where the debrancher is required, but it can't go any further. And so these patients have a substantially milder disease. They have glycogen storage disease. Oftentimes in early childhood, they may look a little more severe, but this tends to be a milder disease that gets substantially better. Okay. Now, this is a child with, let's see what the Roman numeral is. I don't have the Roman numeral. I think it's type 4. Yeah, it's type 4. This is a child with brancher deficiency. He's not able to build branches on his glycogen. Okay? So what happens is that this patient makes a glycogen molecule which is unbranched. So what would the linkages be if I gave you his entire glycogen molecule is large, unbranched, is entirely 1,4, and it turns out to be a rather insoluble molecule, and that molecule accumulates in the cytoplasm of the hepatocyte, and it sort of gets treated like a foreign body and creates a foreign body reaction in those cells, and these patients develop cirrhosis, and they usually die by the time they're 5 from their destructive liver disease. So it's very different. They have brancher disease. Yes?
Liver transplants, would they help with the normal liver supply?
They, in fact, are candidates for liver transplants, but this is, and that's a good question, but this does, in fact, affect other organs, and there's some questions about whether there are going to be other manifestations that appear later, but that is, in fact, the treatment of this disease right now. It's a good question. I'm glad you brought it up. That's the only treatment that we have for these patients. Okay. This child has Roman numeral 2. That's called Pompeii's disease, after the person who described it. Now, this is because these children are unable to destroy the glycogen that accumulates in their lysosomes. A relatively tiny amount of our glycogen gets partitioned off and accumulates in our lysosomes. And we have in those lysosomes an alpha-1,4 glucosidase. It's actually 1,4-M16, if I remember it correctly, which can destroy that glycogen and mobilize it. It's that lysosomal destruction of glycogen, or turnover of glycogen, has no metabolic significance. We can't use it for generating blood glucose. It's a small amount, but it's absolutely indispensable, because when we don't have that enzyme when it's completely absent, by 3 to 5 months we come into the hospital with a severe muscle disease like this little lad here, and if you let that arm go, it would flop. These are the most floppy children that you'll ever see. They have complete muscle hypotonia. This shows you the heart of that patient on X-ray, and their heart is also full of glycogen and lysosomal glycogen, and almost all of these patients die by the age of a year. They have a complete deficiency of the lysosomal glucosidase that breaks down glycogen. Now, occasionally, a patient will come in late in life. This is a 42-year-old male. And I remember I had a woman who was about a 35-year-old black woman who came into the clinic at Washington U, and she just said, "I'm running out of gas. I can't go upstairs anymore to get to my work." She had, as this patient, a partial deficiency of the lysosomal enzyme alpha-glucosidase. And those patients develop a late-onset muscle disease that appears late in life. Muscular weakness, it's kind of progressive, and it also involves the heart. And so Dr. Longmore adages would be correct. This does, in fact, shorten life, even when it appears late in life. Okay. So those are some pictures that I thought I would share with you. I think that the things that you should ask, one question I'll ask you is, one question I want you to think about and be prepared to answer later is, I showed you a patient with debrancher deficiency, right? I want you to think about whether or not his debrancher deficiency would affect his ability to get glucose from gluconeogenesis. Okay? Don't answer it now. Just think about it. You might be asked that some other time. Okay. And I think you should also know, because of the thrust of this conference, the things that I didn't cover, which are covered in that handout, particularly in that feedback sheet, the way in which the inability to recover glucose from glycogen affects indirectly our adipose metabolism and our urate metabolism. The urate I wouldn't worry about so much now, because we'll discuss that in much greater detail when we get to the clinical correlation on gout. Okay, any questions? I'm resolved to finish on. Do you have a psychiatry lecture next?
Yeah.
Okay, so I won't. Yeah.
That adult onset one, is that like type 2B or something? Is that like
The question is the adult onset, late onset called type 2B? I'm not honest. I don't remember truthfully whether they subclassify that. We might. Any other questions? Okay. Thanks very much.